All right, so we've gone through glycolysis and we've gone through the Krebs cycle, and we are getting close to the end. Now it's time to talk oxidative phosphorylation. This happens in two major steps. There's the electron transport chain, and then there's chemiosmosis. Good news is, after you've mastered this, it'll come in handy for photosynthesis. So once you know it for one, you know it for the other. Electron transport chain. This occurs in the inner membrane of the mitochondria, and this process is actually going to create a whole bunch of ATP. Chemiosmosis is specifically a process within here that is pumping hydrogen ions across the inner membrane space. So this is where we really get the big payoff from cellular respiration. It's the reason why organisms with a mitochondria can produce with a mitochondria and with oxygen, I should say, can produce way more ATP per unit of glucose than something that doesn't have a mitochondria or is lacking oxygen. Okay, so the electron transport chain. If you, in a minute, just pause this and try to read this diagram, it says free energy relative to oxygen, the oxygen in O2. And so what it's saying here is if we compare the amount of energy that's available in each of these products, you'll see we start out really high here with NADH. So there's a lot of energy available in this bond that's joining the hydrogen to this NAD molecule. So since there's so much energy available, it's relatively easy for it to split off this electron. And then this electron is going to actually reduce each of these proteins. You see it's getting passed from protein to protein. You see that FADH is also donating its electron. And so by going down this way, it's saying there's less free energy available. In other words, it's kind of like rolling downhill. So we started out with something way far uphill, and now each time it gets passed from one thing to another, it goes further and further downhill. So the electron transport chain gets its name because there's a chain of reactions here that are passing an electron. They're transporting it. And in doing that, eventually the electron lands on these oxygen molecules and forms water. So the electron transport chain itself is the collection of proteins. And they're tightly bound together in the membrane. And they alternate between being oxidized and reduced. So as they catch an electron, so to speak, they have been reduced. As they give it up, they have been oxidized. This process itself doesn't make ATP directly. However, it is solely responsible for taking this electron right here. Now, we totally need this process to take this electron right here and use it to eventually create ATP. This whole process produces water, and another key point here, guys, without oxygen, without this key player right here, this is the whole reason why you need to breathe. Without this whole thing does not work. So if you've got a cell that no longer has access to oxygen and it doesn't have something else it can use to be the final electron acceptor, then this process will not work. It cannot make energy this way. All right, so here's what's going on in here. If we break this down, we're inside the mitochondria now, and we're looking, this side's the mitochondrial matrix. This is the intermembrane space. And so on the previous diagram, we were going from top to bottom. Now we have flipped it. We're going from bottom to top. So NADH has reduced this protein. That pays for, in other words, that electron being moved, that pays for hydrogen being pumped across this protein right here. This protein is a hydrogen pump. So the electron has been passed from this protein onto this one. FADH is dropping off another electron to give this more energy, a second boost of energy. That helps pump another hydrogen into this inner membrane space. Eventually, it falls down to this molecule right here, which you can see is getting transported out. Or I'm sorry, not the molecule, but the electron is getting transported out. And eventually, it's going to hook up with oxygen right here and form water. So the hydrogens now, remember we were following the flow of matter and energy here? Well, matter, the carbon that came into this process is already gone. Now we're just following the energy. And the energy is in the form of these electrons getting passed from one thing to another. So this buildup has been responsible for adding in a bunch of these hydrogen ions. And now that they are added into this inner membrane space, you see there's a much higher concentration of them out here in the inner membrane space than there is down here in the matrix. So these want to go from high to low. And this really cool protein actually lets them do that. So this is showing you a model, a simplified model of what they look like, but it actually does look like a little cellular machine. So this process is called chemiosmosis. It's also referred to as a proton motive force. So it uses protons to actually perform work. The molecule right here, this blue thing, blue and purple thing here, this is ATP synthase. It's an enzyme that makes ATP. So the hydrogens, we paid all this electron money to pump hydrogens into this inner membrane space. Why did we have to use energy? Well, they were already pretty crammed in there. They don't really want to be in there next to each other. They're charged, so they're taking up a lot of space. They're bumping into each other. They're just trying to get out. They can't go through the cell membrane because of these phospholipid tails, which are going to prevent it. So they don't have a choice. Instead, they're going to go through this, and it's shown you in this diagram. It actually spins around before it gets let into the mitochondrial matrix. And this actual spinning forces a phosphate to combine onto ADP, and that makes ATP. Here's a diagram of the entire system here. So this does a good job of breaking down what these two parts are. So electron transport chain, that's the NADH and FADH donating their electrons to pump hydrogens into the space. Now that the hydrogens are in the space, they flow through ATP synthase, and that part's called chemiosmosis. That's the part right there that is responsible for actually creating ATP. Why do we not make nearly as much ATP when we don't have oxygen? Well, right here, oxygen is critical for this step. Without this step, we can't pump hydrogens into that space, so we have nothing to make this pump run. Okay, so oxidative phosphorylation is just another way to break it down. Uses chemiosmosis. Chemiosmosis generates ATP. ATP uses up redox reactions of the electron transport chain, in which electrons are passed down energy levels to a final electron acceptor. Okay, I'm going to put the rest of this schematic up on here. Full disclosure, I stole these notes from someone else. I'm not a big fan of this diagram, but I might take another look back at it and look at it later. If it helps you out, then that's fantastic. If it doesn't, then skip past it. Okay, so big picture from all of this, if you go back through, you'll be expected to be familiar with the amount of ATP that's made relative in each step. So take a look. Glycolysis, we ended up making a net of 2 ATP. Citric acid cycle and pyruvate oxidation, we made another net of 2 ATP. And then oxidative phosphorylation, we made a net of 26 to 28 ATP. There's a range here because it depends on how efficient the reaction is. That's my understanding anyway. Along the way, if we wanted to follow the energy, we saw that we started with glucose, and the energy in its bonds got transferred to NADH and also to FADH2 along the way. Those get passed to this final step and used to pump hydrogens into the inner membrane space, which I'm highlighting right now. That's this area right in here. As they flow back through, the energy eventually gets locked into ATP. Okay. The last part on here is talking about what can happen with or without oxygen.